Effectivement, la conférence sera enregistrée. Donc, je suis Nathalie Isabelle, je suis chercheure scientifique en génomique forestière et environnementale de la division de biologie forestière du Centre de foresterie des Laurentides. Bienvenue à ce deuxième webinaire de la 36e saison des colloques du SCF-CFL. La conférence d'aujourd'hui portera sur les technologies intégrées de soutien à la biodiversité des arbres forestiers et la génomique de conservation. Et je dirais, s'applique vraiment de façon plus large à tous les organismes vivants. Et cette conférence est diffusée aux intervenants du secteur forestier simultanément dans plusieurs sites à travers le Québec principalement et bien entendu des autres provinces puisque j'ai des collègues dont j'ai vu justement le nom s'afficher. Donc aujourd'hui, c'est vraiment avec un immense plaisir que je vous présente la conférencière Dr. Jill Wiggerseen, professeure associée au département d'écologie et de biologie évolutive de l'Université du Connecticut. Jill est également directrice du Centre de biologie informatique et de l'Institut de génomique des systèmes. So it's my great pleasure today to introduce you Dr. Jill Wiggerseen, who is an associate professor of ecology and evolutionary biology at the University of Connecticut. She's also director of the computational biology core within the Institute for System Genomics. Jill a étudié la génétique et l'informatique à l'Université de Californie à Davis, puis la science informatique à l'Université de Californie à San Diego et à la Claremont Graduate School. Ses travaux portent sur l'analyse informatique et le développement de logiciels pour les espèces végétales non modèles, telles les arbres. Donc, elle développe des approches pour faciliter la vie des gens et des nuls en informatique pour les chercheurs en génomique. Donc, Jill est également responsable de la base de données TreeGene, qui représente une ressource communautaire génomique et phénomique intégrée pour la santé des forêts et la sélection. Jill est de la nouvelle génération des chercheurs, c'est une leader, c'est une des personnes que je connais qui est la plus accessible et c'est avec elle que j'ai mieux compris en quoi consistait la bioinformatique. Elle rend vraiment les choses compliquées, simples. Donc, Jill's work focus on computational analysis and software development for non-model plant species, and she facilitates the life of tree genomicists like me. And she also leads the TreeGenes database, which represents an integrated genomic and phenomic community resources for forest health and breeding. Plus personnellement, Jill aurait aimé être vétérinaire pendant ses études graduées à San Diego. Elle a même eu une petite entreprise de gardiennage d'animaux très populaire qu'elle a revendue peut-être pour financer son PhD. Jill a toujours 2 chats, 2 chiens et 2 lapins. Donc, elle aime vraiment les animaux. En plus, j'ai appris qu'un de ses hobbies, je ne pensais pas qu'elle avait le temps d'en avoir, c'est d'organiser ou d'aider à organiser un festival d'art annuel qui se tient dans le désert du Nevada, qui est connu sous le nom de Burning Man. Donc, moi, j'aimerais un jour aller, j'ai découvert ce festival-là, j'espère pouvoir y aller un jour. Donc, ça me fait plaisir encore de recevoir Jill et je vous souhaite un bon colloque. So I let Jill's talk. So welcome, Jill.
Thank you very much, Natalie, for that introduction. And it's really great to be with everyone today. I wish I could be there in person, but hopefully, maybe we'll get there in the near future. So with that, I'm going to talk to you guys about a lot of different things. A lot of it will revolve around thinking about data that we work with, but it will be very related to everything that we're also thinking about regarding forest health. And so I don't think I have to tell or remind anyone here that climate change is going to be with us for some time. It's a current challenge and one that we will continue to face. Regardless of the baseline that we use or the measurement methods that we employ, we're seeing increasing temperatures and more frequent climate anomalies. And just looking at July in this past year, As everyone is very well aware, we saw increased drought, we see increased fires, sea level rise, and a host of other climate anomalies that we're going to continue to face. Related to that, directly responsible ourselves, we're now in the midst of a sixth mass extinction, also known as the Anthropocene extinction. And this also affects, as we hear about often, many of our animal species, but also is going to be dramatically impacting our forests as well. And so of great concern, of course, to Canada is the boreal forest health. And it's been noted that the boreal biome is expected to experience the largest increase in temperatures of all forest biomes. And I know that's not news to anyone here as well. And so we have a lot of challenges in the future related to forest health. And so on the side of genomics, our group is very interested in looking at conservation and biodiversity genomics as a way to try to mitigate some of the threats that are already taking place in our forests. And one of the benefits that we do have, or one of the advantages that we do have, is data. And we have a lot of data available to us today. And so I pull from sort of the field of data science particularly, and think about how we define big data today. And it's actually often defined according to 4 categories, or what we call the 4 V's. So big data comes in the form of high velocity, where, as it sounds, we're just getting a lot of information all the time. And that information is often streaming. And so that information is coming possibly through remote sensing techniques or other ways where we're truly just getting a large amount of data and related to that volume. Huge amounts of data that we, in many cases, really don't have the ability to analyze at the frequency and the amount that they're coming. In addition to that, we have a huge variety of data types. So when we think about this for things like genomics or environmental data, we have even within genomics, we have a tremendous diversity of data types coming from different sequencing platforms. We have environmental data coming in a lot of different forms. And that variety of data, in addition to the volume and the velocity, give us independent and challenges in dealing with that. And finally, the nature of dealing with big data means that veracity becomes a challenge as well. All of the different ways that we're measuring sequence information, environmental information, trait information, means that that information is going to be coming to us at varying levels of reliability. So there's going to be some type of error involved in a lot of the high throughput techniques that we're using to measure this information. And that is an additional challenge, but it's also a benefit. Having this information means that in the right context, we can help make decisions, potentially management decisions as well. And so when I think about big data, a lot of the time we're thinking now about all of these omics that we hear about. Genomics, increasingly epigenomics, looking at methylation, transcriptomics, looking at what's being expressed, proteomics in terms of what's translated in a genome, and then beyond that, metabolomics, so looking at small molecules, and also phenomics, which is now thinking about high throughput phenotyping. And all of these are applying to our forest trees today. And these are all different types of data that come at us at different volumes, in different formats, and also with different air profiles. And trying to integrate across all of these different types of information presents new challenges to us as well. But it also gives us a much more whole picture of what's happening at the organismal level. And so when we think about sequence data and we think about sequencing and traits, that's only a part of the picture when it comes to what we're looking at for our forests. And so environmental data is also a very critical component to that. And so we're increasingly have access to remote sensing data, as well as phenology data that is coming again, being streamed and being constantly monitored and able to be integrated into platforms in order to make assessments. In addition to that, we are also seeing phenomics using and taking advantage of LiDAR. This is a great and tremendous tool for the forestry community in terms of being able to assess a lot of aspects of forest health, species, and measure a lot of the traits of interest in an area and how these are changing over time. And in addition to more of these high-tech approaches that we also have individual citizen scientists that are more enabled to assist on the ground or on the ground phenotyping. And so we have tools that are out there. This is one example of such a tool called TreeSnap. And these enable individuals to help identify, say, invasive species or pests and pathogens as well. And from a scientific perspective, there is also error involved in these types of data collection as well, coming from non-experts in trying to identify these aspects. However, this data is also yet another tool where scientists are limited in their ability to get to all of the places at once. And so this is also another type of data that we can integrate and learn from in our decision-making process. And so when we think about conservation genomics, we know that there is a conservation genomics gap. And that is essentially that there's a huge disparity between what's happening in terms of research labs and generating resources and how well those resources are actually able to be implemented on the other side. And a lot of that has to do with communication, it has to do with training, it has to do with access to resources in order to actually obtain the information we need for the species of value. But it also has to do with how complicated this process remains. And so everything from when we think about, say, the biomedical community, where CRISPR and other tools are readily available and used, they have a single reference genome, and that reference genome is pretty well resolved, and there's a lot of resources that go towards looking at that and the associated models for the biomedical community. When we look at trees, there's still very many species we know very little about, and especially some of the species of great concern at the moment. And so when we think about sort of the life cycle of trying to even get started with that process, we have to start first with generating a high quality reference genome. And when it comes to plants, this process may not be particularly easy. From there, we have to move into generating population scale assessments. And then from there, of being able to evaluate adaptive potential. And then from there, potentially implementing genetic tools into management plans. So that will often involve long-term genetic monitoring and potentially result in decisions around, say, translocation or restoration that can hopefully be facilitated and move faster with this information available. But that upfront time investment is very significant. And so we're very interested in trying to improve that process, get information available to end users faster, and get that information available in the highest possible quality. And so today I'm going to talk about really the data and the technology side of what we do in terms of trying to develop high quality resources for a lot of our forest trees, and then also some of the downstream software that we use in order to integrate across these different data types that I talked about that are coming at tremendous volume and in different formats and with different air profiles. So I'm going to talk about challenging tree genomes. I'm going to talk about the software development that we have for dealing with these complex plants at the genomic level. And then I'll move into talking a bit more about that integration, so integrating beyond genomics and the cyber infrastructure that we use to enable that integration. And so I always like to start with conifers because they're one of my favorites and also very important to your part of the world as well. And so I'm here comparing a couple of the recent conifer genomes that we've completed, giant sequoia and coast redwood, in comparison to other genome sizes. And just a reminder, as many of you know, conifer genomes are very large. And compared to several of our angiosperm tree species, but not all, there's some large examples there as well, they still rank very high. I put the salamander up here because this is another example of a very large genome that's out as well. And as we often are already aware, a lot of our plant genomes are more complex when they're compared to vertebrate genomes as well. And so in this view here, we have a measure of repetitiveness. We have our plants shown in red, and some of the sample model vertebrate genomes shown in blue. A lot of our mammalian genomes sit around that 2 to 3 gigabases in size, and we know that our plant genomes can get much, much larger. We have wheat represented here as the largest genome. And as I just showed before, many of our conifer genomes can actually range somewhere between, 20 gigabases and larger in size. And so this is definitely representative of that complexity, and those large genome sizes are really made up of a tremendous amount of repetitive content. And so way back when we started working on conifer genomes, there were 2 pine species completed around that time, the Douglas fir genome and the 2 spruce genomes, so Norway spruce and white spruce. And these genomes were all completed around the same time. They used what we would now consider to be early high throughput technologies, but they paved the way for a lot of the genome assemblers that we use today, and also developed and helped us develop where we needed to work on new techniques and new technologies in order to actually put these together in a more high quality way. We talk about all the challenges and disadvantages of working in conifers, the large and complex genomes. But we do have that one advantage, and that is working and having the ability to put together haploid assemblies in most cases when we can get enough material. And when we started this process of sequencing and assembling our conifer genomes, like I said, we mostly had available to us what we would consider to be early high throughput sequencing technology, which in this case is Illumina short read data. That's still an important sequencing output today, used and will continue to be for quite some time, I suspect. But when it comes to our very large and complex genomes, it was a challenge, both in terms of volume, coming back to that volume of data challenge once again, not so much regarding error profile, but more a challenge with dealing with, say, really long repeat elements and having a large number of repeat elements and very short reads that are paired with them. So when you think about how you go about assembling a genome, this process has largely not changed over time. We take the genome, we cut it up in pieces, we sequence those as deep as we think we need to obtain the coverage necessary to assemble them. And then we move those shorter fragments into longer fragments by looking at overlaps. We call those contigs. And then we pull those contigs together into scaffolds from that point. There's a lot of different technologies we can use now to move through this process. But when we started, everything we worked on was Illumina short read data and paired data, which is why we kind of see those arrows going in on each side. And we had a lot of it. And so one of the major challenges of moving into genome assembly was the fact that we had billions and billions of reads. And at the time, none of the sequence assembly algorithms were really sufficient to try to put all of this into one place and produce a high quality assembly on the other side. And so many groups were working on this problem at the same time. And one of the assemblers that I worked on developing with Alexi Zimin at University of Maryland was the Mazurka assembler. And the goal of this assembler is to move that tremendous amount of short read data, and it moves those individual short reads and compresses them into what are known as super reads. And then it moves those super reads into assemblies from there. So moving from those super reads into contigs, and then being able to form, hopefully, scaffolds at that point. Now, when Mazurka was first developed, it was primarily involved in dealing with very deep Illumina data and trying to decompress that depth into the super reads. And since then, we've been working on Mazurka to extend beyond that and be able to work into hybrid assembly mode and use some of our newer sequencing technologies as well, which I'll talk about next. One of the disadvantages of working in this system is simply a long runtime and the fact that it still uses a tremendous amount of high performance computing resources. So basically a high amount of memory requiring one of these computers you might see in the background. Most assemblers do that, but Mazurka particularly needs quite a few resources for this task. And so it continues to be a program that is developed and continues to be an excellent choice for working with short read and now also long read data, whether that take the form of PacBio or Oxford Nanopore data. And so when we put together these first genome assemblies, they were very fragmented. Because the primary inputs to these assemblers were short read data, despite spending a lot of time working on a more accurate assembler, we had everything in a lot of pieces. When we talk about how we measure these genomes, the N50 represents the length of about half of the scaffolds in this case. And then the number of scaffolds over the length of 1 kb are listed here. So you can see that we're a long way away from, say, 12 or 13 chromosomes in these assemblies. We have millions, but somewhere between thousands and millions of pieces that they're still in. Now, these resources still have tremendous value to the community and can answer lots and lots of questions, but they are limiting in certain other aspects. And you can see over here when we talked about the sequence input types that were involved, that these are predominantly Illumina, but they have had in some cases some additional scaffolding or low coverage amounts of long read data that contributed. Currently, we're actually working on improving the sugar pine genome, of which we've incorporated long read data into that process. This reflects kind of the statistics going in, and one of the challenges that we're hoping to address is the fact that we're still missing a bit of the genome. So right now, the latest assembly is about 21 gigabases, and we're hoping to get to that 30 gigabase mark. And so when we talk about what it means to be a complete genome and how many genomes out there are truly complete, we can head over to sort of public repositories, things like GenBank, to get an idea of where genomes are today. And so we have about a little over 600 land plants that are publicly available today. About 216 of these are considered to be chromosome scale assembly, and that has doubled. And that is going to continue to increase because we're getting better at putting together genomes. Sequencing technologies are getting better, they are less expensive, and we're able to put together higher quality genomes as a result. But we still have a tremendous number of genomes that fall into this category of scaffold, which means they're just contigs into larger pieces, or even contig level assemblies that sit at that earlier stage. So given the diversity of plant life that we know exists, we still have relatively few genomes that are out there, but we also know that that's changing very rapidly. And one of the reasons this is happening is because our sequencing technologies are allowing us to do that, both in terms of what they're capable of and also in terms of cost. This is a very rapidly evolving field. It takes, if you just look away for a couple minutes, something new will happen with sequencing technologies in terms of error rates or read length or cost. But our current players, primarily in genome assembly, are still the Illumina platform that came out in 2005, and also the longer read platforms that are out today, so Oxford Nanopore and the PacBio platform. Oxford Nanopore is pushing currently on length, and so the ability to generate tremendously long reads. Sometimes they've got reads over 2 megabases in length from a single run, so very incredible. When the DNA is right and all the situations line up, they can get these very, very long reads, and they continue to improve upon that. The PacBio technology has gone the other way and focused on reducing error rate by sort of keeping a 10 or 15 to 20 kb range, and then using multiple passes in order to reduce that error rate almost to something comparable to Illumina, which is very low. And so these are the combinations of technologies that we kind of put together in different types of recipes to handle varying complexities of our plant genomes. And the goal is to do this as efficiently as possible. When we put together some of those conifer genomes, it's still going to take us a couple months to assemble them given their size, but we're getting much better at being able to do this with greater accuracy and greater contiguity. So here is a view here, just in terms of sort of the array of some of the plant genomes that are out and in 2018. And you can see here the different technologies that were used as the primary source of some of those assemblies. Obviously, Sanger sequencing of our original long read, high accurate, but very costly and not so high throughput technology still puts together a pretty good genome. But in addition to that, you can kind of see that our Illumina genome shown here in yellow are on the small side of that N50 value. And so that means they're more fragmented. And as we move in this direction, we see that some of our long read contribution genomes with PacBio and Nanopore are much more contiguous. And so our goal is to implement this in a lot of the plant genomes that we're putting together today. And so there isn't one recipe for success when it comes to genome assemblies. And I just always want to mention that once you move kind of from, say, a hybrid assembly of working with long reads and maybe short reads, you usually still have to do a bit more, maybe medium to a lot more work in order to get to a chromosome scale assembly. And that's where all of these technologies, say, around optical mapping and Hi-C and genetic maps still have a huge role in moving an assembly that's getting pretty contiguous into those final stages of pulling things together at the chromosome scale. And so here's just a note on one of the technologies that I've been working with a great deal. So this is the Oxford Nanopore technology, which from a cost perspective and also a length perspective, as I mentioned before, Oxford Nanopore is trying to push that envelope in terms of length and also come at it from the least amount of cost. This is an ideal platform for tackling some of our conifer genomes and some of the challenges there. So in just looking at some of the more recent conifer genomes that have become available to us, I wanted to show, related to our conifers here, that in using the nanopore technology, the giant sequoia genome that was just recently assembled is about 8 gigabases in size. So it's one of the smallest conifers we work with, but it has been successfully assembled into 8 chromosomes, which is very exciting. And this was done with a combination of Illumina and Nanopore, so that long read sequencing, and then scaffolded with Hi-C. And we did sort of a similar approach with Coast Redwood. However, this genome is one of the few conifers that's a hexaploid, so it did not prove to be quite as helpful. And in this case, we still have more work to do to get us to even more contiguous values. But you can kind of still compare that over here, where we have Illumina-based starting assemblies that will for sure be going through further stages of scaffolding and more sequencing in the future. But these are reporting what we would expect. So over 1 million pieces based on using short read data alone. So algorithms can help, they can bring things together, but they definitely can't perform magic. We need long reads to help us span these challenging conifer genomes. So with this information, with the sequencing technologies we're using, with the new assembly software that's developed with Mazurka, we essentially have a new recipe for these giant genomes. But that's just one challenge when it comes to plant genomes. One of the other challenges, say when you don't have megagametophyte tissue to work with for a plant genome, is heterozygosity. And this presents numerous hurdles when it comes to trying to resolve a genome, regardless of how large it is. And some of the species that we're interested in and are looking at for various conservation concerns relates to our species that have smaller genomes, but in this case, larger heterozygosity. So they provide challenges in and of themselves to work with. But I wanted to highlight another species that would definitely be of interest in Canada, and that is sugar maple. And so we've just recently completed both the sugar maple genome and also box elder. Sugar maple is a slightly larger genome. It's about 600 megabases in size. It has more GC content, more repeat content. It has more expanded gene families when it's compared to Ngundo, which is pretty phylogenetically distant, but also a much more compact small genome, fewer repeat elements in the genome, and more contracted gene families throughout. And so you may be aware that sugar maple is suffering from this phenomenon kind of generally thought of as maple decline throughout its range. And compared to Ngundo, it's a species that does well in very challenging conditions. It's also quite invasive in many parts of the world as well, outside of its native range. And so we are comparing kind of what may be responsible for some of this decline in the maple genomes and what this looks like on the side of sugar maple in particular. Oops. And so these genomes, both are relatively small. Sugar maple is much larger at about 600 megabases, and box elder is just around 400 megabases in size. But this is an example of a genome, despite having some heterozygosity challenges, that with the combination of the long reads and the supplemental Hi-C scaffolding steps was able to be resolved into a 13 chromosome assembly. And so that's shown here with sort of a traditional Hi-C plot, which allows you to see those chromosomes scaffolded together. And so now this is a chromosome scale assembly that we can then take and ask questions of. We can look at things like gene family evolution, we can look at differential expression, and move forward from there. And so this work has been conducted by Susan McAvoy, who's a PhD student in my group. And so after assembling and annotating the genome, she then looked at responses to stress using transcriptomics in replicated plots. And you're not going to be able to follow what's going on this network diagram. But the goal was to try to get an understanding of whether in these particular plots where there were aluminum amendments to replicate acid rain deposition, whether there's an actual difference in how sugar maple responds to that given its decline in those regions. And in examining a lot of the genes that were responsible for aluminum response, for example, it was not actually found that there were a lot of differences or specific differences that could be identified. Given a much larger scale study would be needed to really dive into all of these. But what it seemed like is the challenges of heavy metals and other threats were actually limiting calcium uptake. And that was the primary challenge that we think a lot of these trees are dealing with, given what we were able to see in the genome. And these are just an example of sort of early views of what we can start to look at in these genomes relative to some of the challenges they're facing. This is just step one, looking at a single individual and looking at a differential expression study among a small number of individuals. This still would require looking across a population and really being able to ask further questions, but it is a starting point to start to answer what's going on. And absolutely, in this case, requires a genome to do that. Other species that we're working on as well includes ash species in general throughout North America. As again, everyone is likely well aware, emerald ash borer is a tremendous threat to just about every species right now in North America. It's moving rapidly, and there's very, very little natural resistance. And in the case of natural resistance, this is defined as what is known as a lingering phenotype, meaning that after the impact of emerald ash borer, the tree may live longer, but most eventually do succumb to emerald ash borer impacts during their lifetime. And so through a large and tremendously growing collaboration of folks working on this, including Natalie and Armand, who are here with us today, we're interested in trying to understand the genetic mechanisms of this resistance. And members of this team are looking at aspects from metabolomics to transcriptomics. And Meg Staten in that group has just published the green ash genome and identified some regions that may be responsible for some of this resistance. But we have a lot of work to do here and a lot more sequencing that would be necessary across the range in order to understand where there may be resistance in these genomes. And so that is sort of our pathway moving forward to try to address these questions. In addition, we have another project that we're working on now, going back to our large and complex genomes, and that is eastern hemlock. This is another species I thought would speak to everyone here. Hemlock woolly adelgid has been impacting eastern hemlock through a lot of its range. This is another tremendous challenge for this species, as well as Carolina hemlock, which has a very isolated distribution in the southern United States. Both of these species are very susceptible to this introduced pest. And we do know that there is some natural resistance in the population, but we don't have a good handle on what that is. And so again, the first step of this process is to generate a high quality genome. And Vidya, my PhD student in my group, is starting on that process right now and generating the first resources for eastern hemlock to try to understand this challenge as well. And in addition to working on the genome, she's also generating resources at other levels, looking at transcriptomics, metabolomics, and metagenomics. Both hemlock and ash are thought to have metabolic profiles that contribute to their resistance. And so focusing on this is going to, I think, be an area that is going to be very important in trying to understand what's going on and how to potentially work towards restoration of these species. But we're a little ways from that. We have to still put everything together and see what's going on. And so when we're thinking about putting together these resources for our forest trees, one of the challenges we also come across, especially for some of the large conifer genomes, is simply that maybe some of these species do not have as much economic value. They're very challenging in terms of getting the resources we need to move quickly to address the challenges that they're facing. There's a few projects and consortiums that are out there right now that are helping get resources to the places that need them. One of these is the Earth Biogenome Project. And though this isn't a funding mechanism, it is an ambitious project that's trying to sequence all of the Earth's eukaryotic biodiversity in the next 10 years. And while that's unlikely to happen, what they are helping with is generating standards and guidelines to sort of move us forward through this process and help decide where some of the priorities and sequencing should be and where some of these active threats are in the world. Another project related to that is actually with Oxford Nanopore themselves, and they've just started this org.1 project where the goal is to support rapid sequencing of any critically endangered species. They're using the IUCN Red List definition of critically endangered, but this has already been helpful for the ash project, for example, and we have several of the ash species that have been sequenced through this long read approach, and we've got a couple others we just put on the list that will hopefully get selected, and that will really help us move things forward because generating those initial genomes are still costly and time consuming, so this can help get us going with generating some of these resources. But I want to mention that assembly is truly just the start of the process, as all of you are well aware. We work on a couple of areas downstream of that. And in particular, one of my favorite areas to work on is genome annotation. Once we have this assembly that we've generated, this high quality assembly that's hopefully a combination of long and short reads and has gone through some post scaffolding to get to chromosome scale, then we want to figure out where the protein coding genes are and also other regulatory elements in the genome, and then ideally what those things are doing in the genome. And so this is also another sort of time-consuming and computationally intensive process. So we're interested in developing tools that make it easier, accessible, and more accurate, and putting those out as open source software for others to use. And so why is this process so hard? Well, if we're talking about conifer genomes, for example, again, they're very, very, very large. They remain very fragmented. Oops. And they have a lot of pseudogenes in the genome as well. And in the case of conifers, they often have these uncharacteristic gene structures where they have introns, for example, that can be very, very long, maybe a megabase or longer in length. And so through our process of working on these complex genomes, we started to develop workflows. There weren't really packages that were available out of the box that would work for conifer genomes. And so we kind of had to do a lot of post filtering. We had to put together a lot of different ways to evaluate models and compare them, and then use that information in order to structurally filter and functionally annotate those models. And so why is this process hard? It's hard in almost all genomes, regardless, not just conifer genomes. And it has to do a lot with the fact that there's always going to be errors in your assembly, that there's also going to be some errors in the material that you're using to find those genes. So say your transcriptomes and other material that serves as a reference. You tend to have gaps and you tend to have some incompleteness on both sides of those resources. And you're also relying in almost all cases on machine learning. So looking at some of the gene content that's been represented in, say, a transcriptome, learning from that, and then applying it to your genome. But all of this process is challenging, and depending on the complexity of the genome, whether you're talking about heterozygosity or ploidy or large genome size, that's going to influence this process in a lot of ways. And so we've worked on this kind of from two different levels. On one side, we worked on developing this workflow, and then we actually started with developing tools that would annotate what comes out of those and use that to feed back into the process and improve the prediction of our gene models. One of those tools is NTAP, and this is specifically focused on non-model transcriptome annotation. And this can actually be applied broadly. It's not just plant specific. And the goal of this pipeline was to make the entire process of, say, transcriptome assembly and annotation, as well as annotating gene models, both efficient and more accurate and fairly easy to run. And so this tool does everything from, say, you want to annotate a transcriptome to you want to annotate some gene models. It's going to integrate across similarity search, gene family analysis, contaminant analysis, if you've got transcriptomes in there with fungal or bacterial contamination, and enrichment. And the whole process runs very quickly. You're able to, say, functionally annotate a set of 30 or 35,000 genes in just a short amount of time. And so that is one of the primary tools that we've used to kind of help feed back onto our structural genes and say, hey, are these predictions real? And fix them from there. It's also a way to actually filter and provide higher quality inputs for the prediction process as well. But it's out as a package that anybody can also use in general in order to annotate a transcriptome or annotate gene models that are predicted. In addition to that, we also have a structural filtering package, and this is known as GFACS. And this package is also used at sort of the end of the workflow for gene prediction. And the goal here is to, again, identify unlikely structural models, fix those models, and then feed back into that system to help improve the training. And so this tool is also a standalone tool that can be used with any genome annotation tool as a way to filter and improve those annotations after the process. And so our current active research right now is working on a project known as EASL. And what EASL is essentially trying to do is improve the entire process upstream before we get to the functional annotation and structural aspects so that those programs can work with higher quality inputs and don't have to do quite as much magic when we get to that point. And so here's sort of the high level view here on the top of what the EASL pipeline is doing. It's trying to work with a combination of primary sequence data as well as secondary structures. So looking at RNA folding and learning from information at that level in order to improve the prediction of gene models and specifically translational start sites. The more complicated version of the model that's doing a lot of the machine learning component is down here. I'm not going to go through the details of that, but already we're seeing a lot of improvement in terms of being able to select the accurate start sites and reduce the number of false positives that come out of these packages. To give some perspective on what the scope of false positives are, in this example here, you can see some of the genomes that we've been working on, including the more recent giant sequoia genome. These are the number of initial genes that are often predicted out of sort of the standard packages that are available. And then through our machine learning pipelines, through sort of preliminary versions of EASL, we're able to reduce these down to more reasonable numbers. And we know that plants generally have somewhere between 25,000 and say 50,000 genes. And so we know that these numbers are not representative. Conifers are not expected to have tremendously more protein coding genes in their genome. So getting to these high quality set of genes is very important for the community so they know what they're working with. So next I want to talk less about genomes and more about moving beyond this and into data integration. And so this is kind of what a lot of our group is working on the other side of what we do. So aside from assembling genomes and developing software to do that, we think about how to present this data, how to integrate this information, and how to make it searchable and accessible to the research community. And so the Tree Genes database, formerly known as Dendrum, for people who worked on it for a long time. Started at UC Davis under David Neal, and I've continued that work working on tree genes today. It covers a wide range of species. We have about 2,000 species in the database right now, and it hosts genomes, genes, genetic maps, SNP information, and trait information as well. And during its long history of different platforms it lived in and different technologies, it's now currently a triple database. And what that means is that it is one of about 40 databases that use this triple platform. And this particular platform is a framework for biological databases that make the process of setting up a database and maintaining a database much easier than developing custom frameworks. So it was a lot of work to move tree genes initially into this framework, but now it has opened us up to an entire community that we work with, and we now participate in developing part of this framework as well. And so I think the easiest way to think about triple is that it has sort of three components to the framework. It has a simplified web front end served out in Drupal, which is a common content management system. It has a database backend that has what we call a generic model organism structure, meaning there's a lot of tables for things that might be shared, genomes, SNPs, genes, things like that, and in a relational format so that people can populate and customize these frameworks relatively easily. And then there's also now an analysis component, which allows users to use data in the database and analyze that. And by that, I mean high throughput sequencing data. So really leveraging current bioinformatic tools to analyze their data. And one of the strengths of Triple is the ability to use modules that other members of the community have developed, or to develop your own modules and share them out to the community. So there's two primary modules that we've developed as part of the Triple ecosystem. One of those is known as CartograPlant, and the other one is related to that, called the Plant PopGen Submit module, and that allows us to bring data into CartograPlant. And I'll talk about these a little bit. So CartograPlant, which actually used to be known as CartograTree and is still primarily forest tree data, is a user-friendly and map-based platform. It's in a web-based environment that's there to facilitate meta-analysis. So basically very much a genes to environment perspective. And so we try to serve a wide range of research interests by integrating this data. So conservation genetics data that might be looking at invasive species or pests and pathogens, plant breeding, which might be interested in looking at resistance in new cultivars for crop species or breeding existing cultivars, evolutionary biology in terms of understanding how plants are evolving and adapting to a changing climate. And so when you go over to the CartograPlant view, you actually come into a map, which shows you the numbers of those georeferenced plant accessions on the map. It's giving you an overview of those counts, and it's giving you a lot of options to work with. And so the goal of CartograPlant is to integrate genetic and phenotypic data for those georeferenced plants and provide this through the visualization platform. And it's also going to enable you to search as well as perform data analysis. So you're going to be able to integrate across with it, or work within a single study, or integrate across multiple studies in order to address your questions. Oops. And so I just wanted to show a few views of the CartograPlant interface. One of the things that we've been working a lot on is integration of environmental information. And so here, for example, we have loaded up a view of forest fragmentation layer that we have imported. We provide information on where this information comes from, when it's been updated. You have the ability to adjust the transparency, load multiple layers, or turn on and off layers in the system. Just a view of those tabs. And so we have a large number now at this point of global and what we call sort of regional layers that are integrated into CartograPlant. And so one of the benefits of this integration is that the metrics behind all of these layers are also integrated into the database, which means for every georeferenced plant that we have on the map, and every new one that gets added, we capture all of the metrics at those locations, store them in the database so that you can query that information out at that location or in the vicinity of that location. And so it's a lot of work goes into just trying to get map layers to cooperate with one another, get them formatted for the map, get them available to users. And so we're always taking requests for what people might want to see that they may not have access to currently. So definitely let us know if there's things you want to see in CartograPlant. So here's just a couple of examples of some of the other map layers in action. Things like biodiversity hotspots, human impact levels. We have soil groups here, for example, and then the ability to actually turn on and off multiple layers in view from here. As I said, visualization is one aspect, right, of these environmental layers, but the ability to pull the metrics at each of these locations is really one of the tremendous values of working in that system. And in addition to map layers associated with all of these various environmental metrics, we also have our range maps that are loaded for many species. And this is an area where we're also taking information from folks who may have layers that they want to get uploaded, and we can host those and have those available here as well. And we have about 4 different places that are contributing this information at this time. So in addition to being able to load and unload different layers for things like world clim and soil and biotic threats, you also have the ability to, as I mentioned, you can look at all the information that is stored in the database for each plant point, but you can also select a point on the map while that layer is loaded and get the value at that location as well. And so our goal with the environmental layers is to try to integrate what is needed by the community and curate that information from public sources. Things like precipitation, climate, land use, fire, soil elevation, also various types of plant inventories are available now. I showed an example of the range maps, and that's an area we're constantly trying to expand. And we're also very interested in getting remote sensing data. And there's lots of challenges in working with these data, but this is something we're starting to do a little bit more and very interested in groups who would find value in having this information loaded and work with this information as well. So I also wanted to talk about where these plants come from, because that is one of the largest and most time-consuming aspects of CartograPlant, is to get all the data in there and get the georeference plants onto the slide. So we provide a few different mechanisms for georeference plant data to be imported. So we can take this information directly from scientists. We do have an integration with mobile applications, so both on the citizen science side and also research-based applications. And then we also get a lot of our information from public repositories as well. And so in terms of our direct submissions from scientists, this is the best. This is where people come in, they submit the data, they get a DOI to go in the journal. They know everything about the study. So they're going to give us the best description of that information and be able to not have us guess about what's happening with that particular study. We also have a team of very dedicated undergraduates who work on curating this information from public resources, things like Dryad, for example, and trying to pull together the supplemental data and the main study and do that on their own. They end up spending a lot of their time sending a lot of emails trying to get information that they can't find from these supplemental files. So if you receive one of those emails, that's probably what they're looking for and trying to help move that study into CartograPlant. And then we are getting also a lot of our trait data outside of the studies from resources like TriDB and BN. And again, I mentioned our mobile applications. And these are sort of considered less valid forms of data coming in the veracity component because we can't validate these things as well. But there's still additional data points that are accessible to researchers, and they know where the source of the data is coming from when they're interacting in that platform. And so one of the primary goals of CartograPlant, as I mentioned, aside from storing georeference plant data with genetic and trait information, is to enable analysis. And so by having all of the metadata associated with that study when it's loaded, so not just the sequencing data, but a bit more about where did the SNP come from? What is the assay that generated the SNP information? What reference genome was used if there was one? All of that information is captured. And that then allows things like questions related to landscape genomics, population structure, association mapping, and diversity estimates. And so those are a lot of the workflows that we're focused on in CartograPlant. Here's just a view of where we're pulling some of that information. So Dryad being one of our primary resources. And then also the triple plant PopGen submit pipeline, which I mentioned before was one of the modules that we developed in Triple. This is the actual module that scientists use to import their study information. And so this is where, as I mentioned, we're collecting everything about the experimental design, the raw data, and how the analysis was performed. The user goes through basically a set of screens in order to upload this information that ideally is asking questions that are specific to the study so it isn't too painful, but getting them through to that data collection step and appropriate level of metadata. One of the things that we spend a good bit of time on is actually trying to standardize the trait names or phenotypes that are measured in the studies. And we do this by using the MyAPI standards to do this through the data submission pipeline. A lot of this is actually handled by the pipeline. We try to look at the information and through basically machine learning techniques, try to determine what type of trait is being measured and suggest that to the user if they're not using one of the names already matched. Then we go into ask questions about units and range of values that are acceptable and things like that so that we can also help validate the utility of that data going in. And so we try to make the system fairly robust to ask a few questions and then present some options and also be flexible in terms of formats and things like that. So we handle a lot of the reformatting on our side. But our goal is to definitely get information from the user directly where possible, because then it has the most value, both to that original researcher and also to the larger community. And so here's just an example of that information, say on the tree gene side, if it's a tree-based study. So you have the ability to sort of get a quick view once everything's submitted, download the actual raw files, see a quick view on the map, and then actually go back out to CartograPlant and see the data there as well. And I'll go past this really quick for mobile applications. But I wanted to mention also related to that, when we're pulling in all of this trait data that's coming in, whether it's coming in through the researcher uploading it directly, you also have the ability to connect when you're going through the upload process and connect out to other trait databases to pull in data there, or give permission to trait data that you may have hosted in those repositories so that CartograPlant can use it as well. So we try to make those connections a little bit easier on that side as well for the user. So once we're back here in our CartograPlant view, I wanted to also mention that in addition to the environmental layers, in addition to being able to view studies, there's the ability to actually go all the way in and look at a specific detailed view of a specific tree accession. So here's an example. You can zoom all the way in on a specific tree. You could see what publication it's associated with, species name, exact georeference coordinates, any trait information or genotypes that are associated with that individual. And that also would give you the ability to add that tree into your study into an analysis as well. Sorry, I flipped past that kind of quick. So you could actually add that information as well. The relationship between looking at CartograPlant and then going back and getting a more detailed view of the study is also supported. So you can go back in and get all that raw data, just as flat files and information there, or you can continue to work with that study data in the CartograPlant framework. And so here is another plant detail view here. And what I wanted to show was the ability to also see it on this side. So you can also take a look at this data in CartograPlant and get the SNP information on this side and look at the study as well as the tree genes view if it is a forest tree. And finally, another zoomed in view here. And I wanted to show that you also have the ability to get onto the map, not just zoom in on specific trees or individual accessions, but you also have the ability to select, say, a polygon and just draw a circle on the map, a range or a group of species that may be associated with the same study or may not be, and pull them into, say, an analysis tool as well. And so our goals, in addition to pulling in the environmental layers, as well as importing our georeference plant data from a variety of sources, is also, as I mentioned, the analytics side. And so once we have this data stored in a relational model in the database, that allows people to query that information out, look at individual trees, individual studies, search by traits, and pull that information in to analysis. We do this through a tool known as Galaxy. The users don't see the Galaxy framework, but this is an open source framework originally developed in Penn State that can host and support several open source bioinformatics tools, many of which are involved in a lot of our population structure, diversity estimates, association mapping, and so on. A lot of those tools are supported here through Galaxy. And so Galaxy essentially on the back end can allow you to set up different types of pipelines and customize those pipelines for data input and output. And so what we've enabled in CartograPlant is the ability, once you're logged in and you've completed a search, say you've looked and you've searched at the study level and you've brought in 4 different studies on the same species, you might have the ability then to determine which environmental variables you want to capture across the study based on all of our integrated environmental layers, and then potentially also look at which traits have been measured across these studies. And we're able to do that because we're using those trait ontologies and mapping those onto those terms, and that's enabling us to set up those searches. And once that's complete, and we've also done similar filtering on the genotype side, then you're able to select what type of workflows that you want to run that are going to be back-ended by Galaxy and then run on actual high-performance computing on the back end. And so that allows us to basically set up more complex pipelines that people can plug into. And one of the areas that we're working on right now is actually putting together a lot more on the visualization side to allow people to say, reduce the dimensionality of their set or remove missing data or things like that, especially related to genotypes, and do that more with visualization tools supported in R versus needing to kind of put in parameters blindly into the process. And so that's an area that we've been focused on as well. And so with that, I just wanted to thank numerous people who are involved in all of these projects. I've named a few as we've gone through, but really there's many more here. Everything that we do is incredibly collaborative because our goal is, of course, to develop tools that are useful, to put together genomes that are of high quality, to ask questions downstream. And so as a result of that, we need a lot of input from other people, and we really value that input as well in terms of making these things better and more useful for the community. And so with that, thank you very much for that. Hopefully not too much data and a little bit of information for forest health too. Thank you.
Merci, Jill. Yeah, it was actually, it was great. And I think that we have you provide interesting results from people who are like just starting to work at the, I would say, with the looking for like for the ASH project with trees and better understand what we need and for the reason why genome sequence are so important. So I would like to say thank you very much for your presentation. So Claire, are you the one who's going to take the lead for the question period? And I realized I received many emails during the presentation. Some people were not able to connect, so I will send them the link to the presentation. Hopefully, it has been registered.
So thanks, Jill. Merci. Bonjour à tous. Je suis Pierre Godbout et j'animerai la période de questions. Vous pouvez tous maintenant activer votre vidéo si vous le désirez. You can all now activate your video if you wish. Alors, comment procéder pour poser une question? Vous pouvez poser votre question dans la langue officielle de votre choix avec l'ordinateur, avec le micro de l'ordinateur, en cliquant à droite de votre nom dans la liste des participants et sélectionnez l'icône main levée. Lorsque je vous nommerai, vous n'aurez qu'à enlever votre mode sourdine en cliquant sur l'icône micro. Après avoir posé votre question, s'il vous plaît, vous remettre en mode sourdine. Nous souhaitons que vous posiez une question à la fois autant pour donner la chance aux conférenciers de bien répondre à votre question et de laisser la chance à tous de poser leurs questions. Vous pouvez toujours en reposer une par la suite. So, how to ask a question? Feel free to ask your question in the official language of your choice. Click to the right of your name in the list of participants and select the N icon. When I name you, you will only need to unmute yourself by clicking on the microphone icon. After asking your question, please mute yourself. We want you to ask one question at a time, both to give the speaker a chance to answer your question and to give everyone a chance to ask their question. Si vous préférez poser votre question par écrit, utilisez la boîte du chat. Sélectionnez l'icône en forme de bulle en bas à droite pour l'activer si ce n'est pas déjà fait. If you prefer to ask your question in writing, use the chat box. Select the bubble icon at the bottom right to activate it if you have not already done so. Alors, nous vous demandons de vous identifier clairement en donnant votre nom et l'organisation que vous représentez. Please give your name and the organization that you represent. Alors, la période de questions est maintenant commencée. Question period is open. Donc, je ferai le trafic avec la boîte de chat et avec les micros. Nathalie.
Je peux poser une première question. Jill, I would like to know, nowadays everybody has its own, I see in my field, mainly in forest ecology, lots of people have their own database, and they develop their database in their corner, and so what would you say to convince them to participate to a project or to a platform like the one you are developing? Because sometimes I do not have enough arguments to convince them. So what would you say to them?
I guess the biggest, and I know it's often complicated because a lot of projects may have a specific data sharing goal right up front. And so there may be an objective to develop that resource. If that's the case, I often recommend that people try to develop it in Triple, just because it does make the process easier, and it also makes it easier to share the data. One of the challenges with databases that are developed off of a specific project is simply that the project ends, right? And usually the funding ends, and then a lot of the databases end up unsupported. And I think even with best intentions, that happens, right? And even for tree genes, it's incredibly complicated to try to keep it going all the time. But I think the biggest challenge is, or probably the biggest piece of advice is to ask, do they have a long-term plan, for maintaining that database after the project ends? And if they do and want to maintain that database, then, and there may be value or things they're curating, then I would recommend to put it into a framework that enables data sharing so that we can share that data with them and work in that way. But I think, the most convincing one is like, what happens when the project ends, right? Because that's often they just stay on until the domain name is paid for. And then unfortunately, all that great data from these projects is sometimes lost. So I think that's probably the biggest one. And if people do have a plan for that, then trying to get it into a framework that enables sharing is probably the most important thing.
Okay, thanks. Armand has a question, I think.
Yes, congratulations, Jill. Very interesting presentation, lots of data, lots of work. As you know, I'm interested in gene expression. I do lots of transcriptome stuff. What will be your biggest recommendation or most important recommendation in terms of the data, in terms of gene expression for specific species, tree species that work under your very large umbrella? Will it be, lots of people are doing, let's say, a tree which is susceptible to insect, they do insect feeding. There's a lot of, different organs, roots, stem, leaves. what is the most important component you need, like time frame? But I mean, we all have different interests in terms of the characteristics and the phenotype we're looking at. But if you have like a minimal tools in the toolbox of the transcriptome component, what will you recommend?
Oh, that's a good question. So are you thinking, so when you say recommend, are you thinking in terms of tools or sort of design in terms
No, I mean, in terms of data,
Yeah.
what is the most important component? Let's say you scratch the surface of specific genes for a large population, or you go deep into different organs. And I don't understand, it depends at the end, what are the question asked? But let's say to
Yeah.
make a database clean and as informative as possible,
Right.
what will be your recommendation because of the long experience you have already?
Sure. No, and of course it's one of those it depends questions, but I think the information that we try to collect is the replicate level of the study. We do collect the tissue type that was used. We collect if there were treatments, right, conducted on certain individuals and what those treatments were. We do collect the time points, right, for the actual extractions or sampling of those components as well. And so obviously what that time is and such and how many tissues are needed is going to depend on what that particular pest or pathogen is that they're looking at. But all of that information we collect. And I would actually say that that's an area that we could do a lot more with the database that we're not doing right now. There's some great examples out there of, I think in the Solanaceae community, they have a really nice tool that actually integrates really well across transcriptomic profiles, right? And they actually show in certain tissue profiles and in response to certain biotic and abiotic challenges, what sets of genes are consistently seen across, these studies. And so, and they have this in really nice visualization platforms as well. And so that's one of the areas that's sort of on our list, but we haven't gotten to that yet. So right now we can search all of that information and you can see it, but you really can't look across studies. And I think that's something that is missing for the community. And because we capture it, we can do it, but we just haven't gotten there yet. So all of that is important in terms of being able to capture it. And actually, I think one of the utilities is if multiple people are, studying this, and there are smaller studies over here, or more extensive studies over there, something that can integrate and give you a view of that would be really helpful. Yeah.
Thank you. Sounds good.
Donc, on va passer à une question de Sébastien Clément.
Hello, Jill. Thanks
Hello.
for the presentation. It was really impressive to see the great variety of data integrated all in one place. My question relates more to the open nature of data. From what I understand, everything that's there is open by nature, right?
That's right. That's right.
What would you say to people who are reluctant to submit their data because they want to publish some articles before? And do you recommend them to just submit the data and you withhold the data until they've published, or what's your view on this?
Yeah. We have a way to do that. So through the CartograPlant PopGen submit pipeline, you have the ability to upload all of the information and you can have that held until publication. So you can simply ask for held until publication, or you can set a date. So kind of similar to how GenBank handles that now, that is supported through the system. So you won't, unfortunately, be able to view it yourself in CartograPlant. That is a feature we'd like to enable so that you can also just sort of work with your own data in that framework. In the meantime, we do have login control, and we do have login control around all the analytics that anybody's doing, and you can sort of save that information in between sessions, but not yet for before the study comes out. So that would be something we can do in the future, but we can hold it and we can keep it behind closed doors until that time.
Great, thanks.
Sure.
On va passer avec une question de Patrick Lenz. Et après ça, on ira sur le chat.
Yes. Hello. Thanks a lot for the talk, Jill. It was very interesting, and I'm always amazed how much data you integrate in these databases like CartograPlant, and made me think of that, yes, as a federal, of course, we have different layers of soils and even forest fires and climate maps and remote sensing data. And as well, actually, with Sebastien, who just asked the last question, we are trying to update a lot of the phenotypic data on different genetic trials and permanent sample plots and so on. And so I just wonder to what degree do you think it makes sense to integrate all phenotypic data that you could imagine on something to a database like CartograPlant? Because I just see the mountain of time that goes into it in formatting and trying to put everything in the right shape.
Yeah, it's a huge it is a huge on both sides. I'd actually say trait data is hard, but sometimes the genetic data is even harder, especially before we have reference genomes available. One of our goals is once the reference genome comes available, remap the SNPs right from everything if we have the right information. On the trait side, we're helped by using the kind of machine learning approaches, right? So the actual workflow that we use allows people to upload it in a few different formats in their spreadsheet. So we try to be very flexible, just tell us what's where. And then we take that in, and then the system, we have a lot of code to kind of try to put things in the right place, and also try to say, this looks like this. Is this what you're measuring? This is the description we have. Is this correct? And allow the user to sort of confirm that information. Usually they're just going to still have to tell us something about units, what they're using as null values. It's tricky, right? The biggest challenge a lot of the time is also just the diverse study designs that people have. We think we've got it all figured out, and then I'm like, oh no, we're not ready for that. We have to go back to the drawing board, which happens. So a lot of the challenges come at that level too, is simply, how, different common gardens, clonal materials. Sometimes things get a little bit more, we've got kind of the basics, but sometimes things get complicated in ways that are difficult, in ways that we know the researcher wants to point something out, but we're having a hard time getting it into that format. So there's definitely still a few studies where they might be on the side of us trying to think about how to do that. But we'd like to say that we're going to try to make the process of bringing in trait information as painless as possible so that it encourages people to do that. But one of the things that we know is necessary in order to make it happen is at the journal level. And so we're starting again to work with journals to say, hey, if this is an association study or a population study, we need to have, all the metadata, and then there's a DOI that goes back out because we're all busy, and right, that's sometimes the easiest way to do that. And it is very hard. It's for our students, right? Or anybody curating stuff after the fact. If anybody's tried to pull information out of a supplemental file, you guys know how hard it can be to get the full picture. Some are great, and some you don't want to guess, right? You're going back to researchers and trying to find information. So I will say that it is an ambitious goal for sure. But we keep learning and trying to make it easier.
Thank you. Il y a une question dans le chat.
Yeah, so it's about the bien try. Data is open. This
not
is
fully open. No, not fully open. So we use from bien, we use range map data from them and species information. And the way it works is, so if you are submitting through your trait information, you can authorize access to trait data in try and bien because the individual researchers own the trait data that they've generated through those databases. And so we have a mechanism for that, and we have access to there is a subset of public data through try, and then there's also through bien, we have also a subset through there. But yes, that is a authorization, and so we use bien as a species and range map database first, and secondarily as a trait database.
So James, you have another question, I see.
Nathalie, sinon on va s'interrompre. Bon. James McLean, voulez-vous préciser votre question? Do you want to precise your question, or is it okay with the answer?
No, that's okay. I know these people very well. Brian's a friend of mine. And I've been trying to encourage them for a decade or more to stop this foolishness, but they won't.
Yeah, it's hard on the trait side, surprisingly so. It's actually much more on the genetic side, but yeah, that's where we have to get permission through the authors. We try the same. Yes.
And Jill, I have another question, if that's okay, Benjamin. Oui? My name is James Macklin. I'm a biodiversity informatician and a botanist. And so I am a data integrator, and I lead a few projects that are big in data integration. Many of the folks that you see here are participating in this, and we have an exciting new one starting soon on climate change and population level change. It's very exciting for us. So we've done, just what everybody does. We have a database, or 5, and we have, trying to decide, I think we're trying to decide what to integrate in the sense of bringing data in that is standardized and keeping it in the house versus a word that an acronym I didn't hear you say, which I think was interesting, is APIs versus, programmable interfaces that allow us to talk to resources, standardize them in the communication pipeline, but not keep them because they belong to others. We want them to version in their own place. We don't need them in our silos, but
Right.
we really want them to be communicating. And of course, our modern stacks, our modern technologies are very service oriented. And I think this is wonderful because it takes away the pain of maintaining a silo. Yeah.
Yeah, absolutely. No, you're absolutely right. I didn't talk about that, but we do that with GenBank data, for example. So we don't actually host the raw data locally. We connect to it if it's available there, and we do this through the triple. So there's a triple exchange mechanism that's essentially an API that means that however everybody has their own database set up is their business, and we can connect and search and connect through other databases as well. And we essentially do that with what we can work with in BN as well. And that's how we talk to them once data is opened up as well.
That's great. Thanks.
I'd like to see that for the person who has a question, please give your name and the organization that you represent. It will help the presenter and ourselves to know where you come from. So, Patrick, do you have a question?
Yes, I just have another question just to introduce myself. I'm Patrick Lenz, working as a scientist in quantitative genetics and forest genomics, and at the Canadian Wood Fiber Centre, which is part of the Canadian Forest Service and Natural Resources Canada. And my main expertise is in actually in genomic breeding tools and other breeding tools to accelerate selections. But I have a little question, and you're not, Jill, don't feel obliged to answer it, but how do you do it to keep an army of people to make the link and with the data? And the question comes a little bit to see what we could also on our side do better, and maybe also help for integrate things in the future with some of your databases, for example.
Sure, that's a good question. It's I would say working with data is always challenging, especially when you're sort of trying to encourage people to share that data. We honestly, I think the biggest advice I have is that some coordination with journals and granting agencies that are more formalized, that it isn't just I will share the data at the end of the project, but that there's a plan and it starts at the beginning of the project, not when funding runs out and everybody's gone, right? Which is often the case, or, sometimes we get a whole bunch of spreadsheets sent to us after the fact, and that can just be overwhelming. So I would say it's hard. We have current funding for Tree Genes right now, which enables us to have undergraduates help with curation, and to have developers work on tools that make it easier. So I'd say the combination of trying to make workflows accessible, but also really working at the other levels of how data needs to be submitted is probably the best advice. Otherwise, it is hard, right? We don't have nearly all the data we would like to have or probably should have. And without researchers submitting on their own, there's no way we could keep up. So I think I'm not sure if that answered your question. but maybe. Cool.
Perfect. Thanks a lot. And
Sure.
thanks a lot for your talk again.
So there's no question in the chat. It's just Nathalie.
Actually, I'm looking for a way. Would it be possible for us in some ways to contribute to your database? For instance, we have data that actually are sitting somewhere, and we've realized that we have a lots of data. Actually, we have a lots of common garden, lots of things. Some of them were characterized, but there is no real, I would say, current research done on that. And sometimes we need to, you know, there might be some way to perhaps to contribute to your database and hire ourselves, perhaps the undergrad. I think it would be for us, especially in forestry, and I would like to expose the undergrad more to you know, the database minding, because I see I'm involved in the on a committee that is there to they call it what is the name in English? We look for, you know, the level of there are a number of requirements to study in forestry, and they have to fulfill some the university has to fulfill some objective with that. And I see that in some case we were mentioned that a lot of in the future forester will need to deal with, a lot with database and data information in real time. So I was wondering if like hiring sometimes some students to do that would be a good way to contribute, because I see your work and perhaps people don't realize because everybody is not in genomics, but you know, initiatives like that are very important. And this is the genomics community is in this position today because people share their data, and this is really important. So I was wondering if there might be some way for us to contribute to your database.
I think that would be excellent. I've found that a lot of students are really eager to learn about how these studies are put together and interact with the tools. And often they actually provide us with some of the best feedback because they're the ones going through all the studies and seeing what, you know, what the actual system supports and what it doesn't. And they're able to give us some feedback and develop from there. So I think that would be a wonderful idea. It just often takes a set of hands and eyes because it's going to be a bunch of spreadsheets, and we need some help getting it in there. I think that would be wonderful.
Okay, especially nowadays that people are sometimes alone. So I think it's yes, we will discuss that later.
Sounds good.
So we still have a few minutes. Is somebody have a question? Yeah, Martin.
Hi, Jill. Martin Williams. I'm a research scientist working on species conservation with the Canadian Forest Service. And I'm just wondering how does the database would deal with, I guess, species at risk information like coordinates or things like that might be at risk in some country, but not in another. So how would that, if data was integrated, how would the database deal with that kind of issue? And would there be a way where coordinates could be, I guess, hidden or exact locations hidden so that
we have that. We actually work with the Global Timber Tracking Network and World Forest ID, and that is a primary concern for a lot of the species there for tracking illegal logging. And for those species, when we put them on the map for the public view, they are what we call it a buffer. They're sort of randomized slightly away from that where they're actually sampled. And then if you're logged in and you're one of the authorized users to actually see that data, then you're able to do that. Now, what we haven't thought about is if it's okay to see it somewhere and it's not okay to see it somewhere else. It's at this point, it's sort of handled on a study by study or submission by submission basis. But I think if it went in like that should be okay. Somebody could, so the way the system is set up right now, you actually can see based on the icon if it's an exact coordinate or if it's an estimated coordinate. So at least the public knows it's correct or it's not correct, but you could keep that public view estimated. Some things are estimated because they're truly estimated and we just don't have it. Some things are estimated intentionally. So yeah.
Perfect. Thank you.
Sure.
Donc, il est maintenant midi, à moins que quelqu'un se manifeste maintenant. Je vais conclure le webinaire.
Okay.
Donc, thank you very much, Jill, for your presentation. Integrated technologies to support forestry, biodiversity and conservation genomics. Alors, en terminant, je vous invite au prochain colloque qui aura lieu jeudi le 13 janvier et qui s'intitule Forêt urbaine et changement climatique. Le conférencier sera Alain Paquette, professeur titulaire au département des sciences biologiques de l'Université du Québec à Montréal. Surveillez également notre fil Twitter et Link pour en savoir plus. Merci à tous de votre participation et bonne fin de journée.
Thank you very much, Jill.
Thank you. Thank you, everyone. Bye-bye.
Bye.
Bye.